Hello everyone, I'm Al and this is H. Today we will discuss about nucleic acids. So what's nucleic acids? Nucleic acids are large biomolecules essential for all known forms of life. They are composed of monomers called nucleotides which contain a phosphate group, a sugar and a nitrogenous base. Nucleic acids carry genetic information and play critical roles in cellular processes. The two main types of nucleic acids are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. First, we are going to talk about DNA, aka deoxyribonucleic acid. First, we are going to look at its structure. DNA is composed of two long strands that form a double helix. Each strand is made of nucleotides, which include a deoxyribosugar sugar, a phosphate group, and one of four nitrogenous bases, which is adenine, thymine, cytosine, and guanine. These strands are anti-parallel, meaning they run in opposite directions. The bases pair through hydrogen bonding, which is adenine with thymine, AT, and cytosine with guanine, CG. Then we are going to look at its function. DNA stores and transmits genetic information. It provides the instruction for the synthesis of proteins, which are crucial for cell function and structure. DNA replication ensures that genetic information is passed from cell to cell and from generation to generation. Next, we are going to look at RNA, aka ribonucleic acid. First, we are going to look at the structure. RNA is typically single-stranded, though it can form complex secondary structures. RNA nucleotides consist of a ribose sugar, a phosphate group, and one of four nitrogenous bases, which is adenine, uracil, cytosine, and guanine. The uracil replaces thymine, which is found in DNA. Next, we are going to look at its types and function. mRNA, aka messenger RNA, carries genetic information from DNA to the ribosome, where proteins are synthesized. tRNA, aka transfer RNA, brings the appropriate amino acids to the ribosome during protein synthesis. Next, rRNA, aka ribosomal RNA, forms the core of the ribosome structure and catalyzes protein synthesis. And lastly, we are going to look at the function of nucleic acid. First, genetic information storage. DNA contains the instruction necessary for the development, functioning, growth, and reproduction of all living organisms and many viruses. Second, protein synthesis. DNA is transcribed into RNA, which is then translated into proteins. Proteins are the workhorses of the cell, carrying out various functions. 3. Gene regulation. Certain types of RNA play a role in regulating gene expression, ensuring that genes are turned on or off as needed. Fourth, catalytic roles. Some RNA molecules, known as ribozymes, have catalytic properties and can facilitate chemical reactions. Nucleic acids are fundamental to the functioning of cells and organisms, carrying the genetic blueprint and playing a key role in the synthesis of proteins and the regulation of gene expression. That's pretty much everything I have to say about this topic. I will now pass the next topic to Hesh.
Good morning everyone, Hesh here. Today I'm going to explain the importance of nucleic acid in a cell. Nucleic acids are critical biological molecules found in all living organisms. They serve multiple important functions within cell. Number 1, they store genetic information. DNA and RNA molecules store instruction for the structure and function of cell and organism. Number 2, they act as catalysts for biochemical reaction. Some RNA molecules, known as ribosomes, possess enzymatic properties, facilitating chemical reaction in cell. Number 3. They participate in protein synthesis. Messenger RNA molecules carry information from DNA to ribosome, where proteins are assembled. You can refer to Figure 4.4 and Figure 4.5. Number 4. They are involved in cell signaling. Certain RNA molecules, called microRNAs, regulate gene replication and cell communication by binding to mRNA and preventing translation into proteins. Number 5. They contribute to cellular energy. ATP, a nucleoside triphosphates, serve as a primary energy currency within cell, providing energy for various metabolic processes. That is all. Thank you.
Thanks, H, for the explanation. That was really interesting. Now I will continue on to the next topic, which is formation of chromosomes from DNA and proteins. The formation of chromosomes from DNA and proteins is a complex process involving multiple levels of organization. The explanation in the textbook is quite simple, so I'm going to give a more detailed explanation here. First, we are going to talk about DNA molecule. I have already talked about it before, but I'm going to repeat it again. DNA, or deoxyribonucleic acid, is a long double-stranded molecule made up of nucleotides. Each nucleotide contains a sugar, a phosphate group, and a nitrogenous base, which is adenine, thymine, and cytosine and or guanine. The sequence of these bases encodes genetic information. 2. Histone. Histones are proteins around which DNA winds. There are 5 main types of histone: H1, H2A, H2B, H3, and H4. A group of 8 histone proteins, 2 each of H2A, H2B, H3, and H4, form a structure called a nucleosome core particle. 3. Nucleosome formation. DNA wraps around the nucleosome core particle approximately 1.65 times, forming a nucleosome. This structure resembles beads on a string, where each bead is a nucleosome and the string is the linker DNA. Histone H1 binds to the linker DNA between nucleosomes, helping to compact the DNA further. 4. 30 nanometer fiber. The nucleosomes coil into a higher order structure known as the 30 nanometer fiber. This fiber forms through interaction between the nucleosomes, creating a more compacted form of chromatin. 5. Loop domains. The 30 nanometer fiber is organized into loop domains that are attached to a scaffold or matrix made of non-histone proteins. These loops help further condense the DNA. 6. Further compaction. During cell division, particularly in mitosis and meiosis, the chromatin undergoes even further compaction. The loop domains fold and condense to form the highly compacted structures known as chromatids. 7. Chromosomes. Each chromosome consists of 2 sister chromatids joined at the region called the centromere. The chromatids are identical copies formed during DNA replication. Chromosomes become visible under a microscope during cell division due to this high level of compaction. In summary, the formation of chromosomes from DNA and proteins involve the wrapping of DNA around histone proteins to form nucleosomes, the further coiling of nucleosomes into the 30 nanometer fiber, the organization of this fiber into loop domains, and the ultimate compaction into the visible chromosomes during cell division. This hierarchical structure ensures that the long DNA molecules are efficiently packed within the cell nucleus while remaining accessible for processes like transcription, replication, and repair. That's all for me, and this is the end of our presentation. Have a good day and goodbye.